Hello everyone, I'm Xu-Heng. Today, me and Dandy are here to talk about the journal article, Higher Energy Collision Activity Dissociation without a dedicated collision cell. This article was written by Graeme, Douglas, Justin, Michael, and Joshua, etc. Basically, this article is about an upgrade of HCD called IHCD, and IHCD is compared to other dissociation for efficiency. High energy C-trap dissociation is a CAD technique specific to orbit-trap mass spectrometers. In HCD, ions pass through the C-shaped trap and enter the HCD's dedicated collision cell, and they can be dissociated with each other. Then the ions are returned to the C trap before being injected into the orbit trap for mass analysis. So we know that HCD requires a specially designed and dedicated collision cell. This technique is very effective for the identification of phosphorylated peptides and is compatible with a quantification based on isobaric tag labeling. Regardless of the advantages, the use of a dedicated collision cell increases the complexity and cost of the instrument, especially for independent QLT system. So the authors introduced an upgrade version of HCD, which is IHCD. It can be performed in the ion injection pathway of a mass spectrometer with a standard atmospheric inlet, rather than dedicated collision cell.
So I'll be talking about the procedures of IHCD. So human embryonic stem cells were used in this experiment, and various enzymes were used on these cells, followed by cell lysis. Then the cells were digested with trypsin and labeled with ITRAQ, which is the type of isobaric tag labeling. Then I will be going through the next step along with this figure. After injection and separation of the specific precursor, the separated precursor group is transferred back along the ion injection pathway with high kinetic energy. When ions enter the high pressure area near the AP inlet, which is the Q0 stage, the ions collide with neutral gas molecules leaking from the atmospheric source. The precursor ions collide with the background gas molecules and fragmentation occurs. The generated products are trapped here, and the ions fragments travel back to the QLT along the ion injection pathway, and the products are now ready for mass analysis. Then liquid chromatography tandem mass spec is done, and the 10 most intense precursors were selected for data-dependent activation using IHCD. The QLT mentioned over here is a quadrupole ion trap that uses dynamic electric fields to trap charged particles. The reason why the authors used this was because the trap designed increased ion storage capacity. It had a faster scanning time and it had simple structures. Before I discuss the results, another technique is used to compare IHCD, which is the CAD. It is a mass spectrometry technique, and in this technique, the selected sample ions are accelerated by applying an electric potential and then collide with neutral molecules, such as helium. In the collision, some of the kinetic energy is converted into internal energy, which results in bond breakage and the fragmentation of the molecular ion into smaller fragments. These fragment ions can then be analyzed by tandem mass spectrometry. The use of ion trap has made it a widely used dissociation method. This chart presents the results of the procedures mentioned on 3 generations of QLT, which is in black line, gray line, and dotted line. It depicts a similar experiment in which we monitor the ejection of product ions from the Q0 stage via the ion injection pathway to the QLT, and it emphasized the efficiency of IHCD regardless of which QLT was used. Then a mass spectra is generated by IHCD, which is in A, and HCD in D, of triple protonated angiotensin cations. We can see that the result of HCD is close to that of IHCD.
In chart A, show the ability of IHCD to identify peptides from a complex mixture. The ability of identification of IHCD is better than that of HCD and is similar to CAD. The performance gap between IHCD and HCD is mainly analyzer driven. IHCD generates more sequence informative MS spectrum than CAD. Taken together, this identification and quantification data indicate that IHCD is an excellent method for isobaric tag-based quantification. In chart B, show that IHCD is capable of identifying and generating quantitative information from isobaric labeled peptides. The reason why the author replaced CAD with PQD is because CAD cannot retain quantitative reporter ions. Compared with PQD or HCD, IHCD produced twice as many recognition results. In summary, this identification and quantification data show that IHCD is an excellent method based on isobaric quantification. Before I introduce this picture, I want to tell you what is a PQD. PQD is a variant of a CAD designed to overcome low quantity cutoffs and produce moderate imine ions levels. So we use PQD to compare with IHCD in the MS spectral pattern. This picture shows the dissociation of an isobaric tag label peptide K9 tested by using PQD and IHCD. In both cases, the resulting MS spectra easily identify the corresponding peptide. However, the isobaric tag reporter ions are tenfold more intense in the IHCD spectrum. The results emphasize the advantage of IHCD. It is robust, sensitive, and is compatible with any MS system as long as it has an AP inlet.
This paper compared CAD with IHCD and provided a similar or better recognition rate, and it has all the advantages related to bean-shaped dissociation. First of all, it's compatible with isobaric marking for multiple quantification. Secondly, we can discover, transfer to the conversion target analysis of the precursor to the product. Third, it eliminates the inherent low quality cutoff value of CAD. And lastly, it improves the sequencing ability, especially for peptides with post-translational modifications. IHCD eliminated the need for a dedicated collision cell, thereby reducing the complexity and cost of the instrument. To conclude, IHCD is an effective and easy to implement technology.
Thank you, everyone.